Okay, it's a real pleasure to start this academic year's seminar series for the Center for Clinical Trials and Evidence Synthesis. I am delighted to have our first speaker for the year, Dan Hanley. As I told Dan yesterday when we chatted, I've known him since the beginning of the last quarter of the last century of the last millennium, which means we've known each other for a long time. I first met Dan when I was an intern in internal medicine and he was a resident. We were both at Cornell at the time. We both then moved to Hopkins. Dan, after completing his internal medicine residency, did a fellowship in neurology, followed by a neurology residency at Hopkins, and then a critical care fellowship at Hopkins. He started the neurology, neurosurgery, intensive care unit, and became an expert in critical care of patients with brain injury. He finally saw the light and moved to clinical trials, and he founded the Brain Injury Outcomes group, and has done multiple clinical trials in this area, including MISTY-3 and CLEAR-3. Subsequent to that, when the NCATS funded the Trials Innovation Center, part of the Institute for Clinical and Translational Research, he headed that. I've had the pleasure to work with Dan on some COVID-19 trials where a couple of our faculty have been involved, and the Trials Innovation Center has really put together the support for managing the trial and done it very effectively. So he's going to talk to us a little bit about what he's done with BIOS and sort of the approach that they use over in BIOS and in the Trials Innovation Center and to managing clinical trials. And I'm delighted to have Dr. Hanley speak today.
Thank you, Doug. I'm going to, what happened there? There we go. That's not good. I'm going to tell the story of the development of the Division of Brain Injury Outcomes into an academic research organization, and our role with Hopkins ICTR in multi-center clinical trials. This will not be what I like to do, which are data-driven studies. This is more of a anecdotal talk, and for those of you who are disappointed by not having enough data, I apologize. I have pulled together some data about clinical trials that I think is informative. This is a pharma slide of what it takes to get to one FDA-approved drug in terms of compounds, time, and different areas of review. And the reason I'm showing it is clinical trials are hard to do if you want to change practice. And if you take a lesson from pharma, which I think we should, since they do pharma and devices do many more trials than we do as individual investigators, you have to be prepared to do sequential trials. Said another way, this is what most of us as academic faculty see in terms of clinical trials. It's long, slow, and lots of blockades. I'm going to put the bibliography first for two reasons. These are interesting background reports that led to a change in NIH's clinical trial approach. And it really starts with two major things: an Institute of Medicine report and a GAO report that said NIH was not doing clinical trials very well, that their clinical trials were remarkably delayed, as was obvious to GAO from large unexpended balances in the trials accounts. That's led to some changes. Oh, I switched. Here we go. Now go forward. Okay. My disclosures. For the last 20 years, I've made my living doing clinical trials, so I probably have biases, and a number of things that we've done are dependent upon the FDA, pharma, and device companies. And I have relationships with a number of professional patient and public and NIH boards. I'm not doing well with the arrows. My youngest son said, if you're going to tell a story like this, you ought to tell a little bit about your family and how you were nurtured. And Doug reviewed this. I'll point out that I'm the oldest of 4 children, which tells you something about probably how I behave. And my whole family was medical, with a father who was a generalist and a sports medicine person, and a mother who was a microbiologist. And for some reason, in that generation, all the kids went into medicine. And I have several burdens above and beyond my personal friendship with Doug Jabs, who was an intern when I knew a lot as someone who was 12 months ahead of him. I also have been, for whatever reason, very interested in how the whole human body functions, and been less of a reductionist. And that carried over into my final fellowship, which was in anesthesia, critical care medicine, where I did systems physiology, but at the molecular level. So I like whole functioning systems, and being in systems physiology, or running an ICU, or running a clinical trial organization, it's the whole system that's been interesting to me. In my first 5 years as a Johns Hopkins faculty, I got exposed to a lot of trial work, and through a very robust group, the National Antiviral Study Group, where over the next 10 years, we did 3 trials and a longitudinal study. I brought the discipline that I learned there from working with very good trialists, where I was both a site, and then I moved to being the neurologic expert in this National Antiviral Study Group. I moved that over into what I was doing in the ICU, as you can see, with longitudinal basilar artery occlusion, myasthenia gravis, subarachnoid hemorrhage trials, and then an experience through a number of pharma trials. And in doing this, I always asked why questions that came from my bench experience. And during this first 20 years of my career, I was working in the ICU and managing 1 to 3 RO1s in the areas on the right-hand side. You may think that's schizophrenic, but the bench is nice because you can design experiments and do a whole set of well-powered experiments in 3 to 6 months. And that gives you enough courage to design an experiment in a trial that may last 5 years. So very important to me, maybe not important to everybody, but important to me. If I were to name this talk, I would say this is a story illustrating the value of sequential trials. Early on in my career, an anesthesiologist, Bob McPherson, said, I know what research is. you search and you search again and you search again, and that's why they call it research. I guess what I've learned in the tiresome part of research is that there's always an alternate hypothesis to be explored. And if I had one piece of wisdom, that's what I would give to everybody who's interested in clinical trials. And the results of your prior trial should inform you to do one of two things: undertake the next trial in that area, or move to a different area. And so there's always something to do if you look at the alternate hypothesis. This is an illustration of all the red areas are areas of my personal interest. And this is an illustration. All the black are clinical trials that we actually have done. How that sequential hypothesis is what we've lived. And we've lived it, if you look at the roots, through the translational or biomarker lens of a number of basic science perspectives. So we're now called the BIOS, or the Brain Injury Outcomes Clinical Trial Coordinating Center. I have two colleagues, Karen Lane and Nicole McBee, who are neurology faculty members, without who the center would never have been developed, and who are both the brains and the muscle behind the center. They've been with the center since the very beginning, and they are the lifeblood of the center, and I'd like to acknowledge that. I do have a little bit more disclaimer at this point. I like Lucy Van Pelt, and a lot of the statements, like the ones I've just made, are psychiatric in nature. Any wisdom that comes out of it is acquired through failure, not necessarily success. It's usually the good advice of Karen and Nicole that keep me out of trouble. But today I'm a neurologist practicing psychiatry, and the presentation contains a lot of bad stuff: aspirations, theory, even worse, social theory, business theory, and anecdotes. This is BIOS in 2018 or early 2019. We're a bit bigger now, but we're organized into areas of expertise, as you can see, and I'll talk about that as we go through the slide set a bit more. We started as a concept in 1999 to find what was actually happening in terms of human performance late after you were admitted to the neurointensive care unit. And as you can see, we gradually acquired an increasing number of clinical trial awards. We had a good year last year with 5 new trials that we're starting. We do a full range of clinical research activities that can support trials, as you can see, both in the methodological and in the project management domain. I'm going to talk more today about the project management domain, or trial execution, and using tools to execute trials, and how we do it. Certainly study design and starting with the right design is important. And then planning, which manuals of procedure, data reporting plannings, the teaching and training about how you want to interrupt current clinical practice is critical as well. So as I started BIOS, I've listed out 6 lessons that I learned. Design is important. And I was lucky enough to have performed, as I told you, multiple well-powered small cohort studies that had short turnaround times to learn how well or how poorly you designed the study. From that, the single most important message that I've always taken as I look at a problem is to define, test, and then retest, and not accept the precision of whatever measurement you're going to use for your primary outcome. And the second thought process is, in clinical diseases, it's not different than the bench. There has to be a causal pathway, and you need to, as best you can, understand the causal pathway while you understand the primary outcome. And for clinicians, I think you have to ask the question, what's the time frame in which your outcome will become stable? Not accept what what others say, but rather determine that from your own data. And once you've done that, then learning to utilize a diverse set of data collection instruments, and to develop within your trial methods for measuring the accuracy and reproducibility of your measurements helps you perform a good trial rather than a not good trial. And the final thing is equipoise, which is the alternate definition of alternate hypothesis. I've come to learn that my thought about the causal pathway is likely to be proven incorrect at least half the time. So in designing the trial and in executing it and in the analysis stages, to always look for the alternative as hard as you're looking for what you believe is the causal pathway. So those were the lessons that we brought to the start of BIOS. Here's an example of at the beginning of BIOS, we did things, and you'll see I was doing the trials while I was in the ICU as well. And this is the beginning of BIOS, which is an FDA Orphan Drug Award. We did them in sequence, and we tried to ask the next question. This was, do people survive if you treat their interventricular hemorrhage? This was a randomized short trial, and then a larger one funded by FDA, a dose finding and a PK, and then a large international phase 3 trial. And we followed the same thing in the MISTY set of trials, which looked at brain hemorrhage in a different location in the brain tissue. That was a set of 10 sequential brain hemorrhage trials that were not subarachnoid hemorrhage, which I'd worked on in the decade before, but rather were bleeding directly into the brain tissue. And we used measurement. Our goal was to decrease the size of the blood clot, with the idea that you'd be much better off if you had a smaller brain bleed than a larger brain bleed. And some people thought that that was trivial, but it turns out that it defines what is clinically important. And the hypothesis that a smaller brain bleed leaves less toxic elements in the brain tissue has turned out to be a robust predicator for both drug development and device development. And we've spawned in the world with these 10 trials, probably another 200 to look at those issues. When I look at what we've done, we've ended up in the same place in both the clear and MISTY paradigms. We are now back after in each, a large worldwide trial, 50 site and 70 site. We are now back to using the measurements that we defined and that we uniquely worked on making accurate and sharing how to make them accurate with the world. I'll show you a bit about that later. We're back to using those measurements to improve per protocol performance. In both trials, we easily demonstrated that a smaller blood clot allows you to survive, but only the per protocol data in our sequence of trials, only the per protocol data showed a benefit and functional outcome. So now we're going back to phase 2, and we're going to help people do the protocol better. That's one of the sequential values of trials. Here's how we executed it. In the red was the percentage of people who achieved the protocol proposed reduction in the phase 2 trial, and in this phase 3, which is clear, and in the MISTY trial. We now have done a effectiveness study in which 90% are achieving the goal, and we're hoping to now take this out to another large randomized trial in the MISTY area, and we're hoping to do the same thing in CLEAR. And this is all based on just measuring the size of the hematoma and using a tool that clinicians were using qualitatively, the CT scan, to make not just a measurement, but we found that most of the world used approximators, and the approximators weren't very accurate to make an accurate measurement. We did that through a program called STRIDES, which the NIH has put out the opportunity to do cloud-based solutions for trial efficiency. And the URL is down there at the bottom. What they did is NIH matched you up with either Google or Amazon, and you took your problem to them, and they helped you develop a cloud-based mechanism for making measurements. In the first brain hemorrhage trials I showed you, we would take a CT scan like the one on the left, and a person would draw the margins or the region of interest for each of these little segments, and they do it on 30 slices of the CT. It would take half an hour to an hour with a lot of variation, but better than the estimators. And then you would add up the full thickness of each slice to get a volume. This would usually occur in our first studies. It occurred after the study was done. Eventually, by our third study, we were doing it in parallel and give the result to the sites the next day. But using this NIH STRIDES program in the Google Cloud with appropriate programming and an artificial intelligence algorithm, we can upload a DICOM file and run it through a de-identifier so that it's everything is HIPAA compliant, and sort the image and make an automated assessment of what those volumes are in that slice, and add them up in essentially 100 seconds, where it would take days or hours beforehand. And then we can return it directly to the site. It's that kind of precision that's allowed us to use volume as a biomarker and make modifiable changes in clinical practice that others will accept, which is very important in running a consortium. And this is the accuracy story. If you just do it with a human and the best programming which is semi-automated. This bland Altman plot with the narrow band for accuracy is breached in both the ICH and the IVH very frequently. But down here in the blue box is machine learning. And I think you're going to see a lot more of this in trials as well as in practice. So the lessons I learned from those 10 trials, 5 years sounds long, but time really matters. And you have to overcome the barriers one at a time in each study and each site. The task ahead is what you have to focus on, not all the hurdles that prior protocol, prior IRB, prior regulatory people asked you to accommodate to. The consortia does the studies, not you as the PI. So spend your time listening to people in the consortia and help them disrupt their practice by doing your study. And in doing that, unfortunately, a lot of people have opinions. Use equipoise, it's your friend. Convince people that they're doing the study because we don't know the answer. And remember, all this time you're spending with people is because trials are disruptive. And you need to use data, not ethics, not opinion. You need to use data to persuade all the experts that are out there. There are commercial people who will attempt to sell you trial services. They're usually overpriced. The rise of the internet and media has made doing many of the things we need to do in trials inexpensive, and you can buy off-the-shelf solutions that are much cheaper than the commercial trial people will offer you. Always integrate your trial communications with the actual actions that you and your sites are performing. Everybody has trouble with trials, which is why NCAT's invested in operationalizing trial innovation. I'm going to run quickly through this. This is the annual spending by pharma, but you can see that it hasn't led to an increase in new drug approvals. And this small increase is perhaps explained by the rise of biologics, which is a whole new set of mechanisms, but there hasn't been a big increase in approvals that matches the amount of spending that's gone on in private pharma. Participants are important to trials, and this is a public attitude about trials from a large survey performed by Ken Getz at the Tufts Center for Drug Discovery. 12,000 individuals, 85% said clinical research is important and we need it to develop new solutions. But 59% were unable to name a place where this research or clinical trials occurred. 90% believed it was generally safe, but 50% of that group said they hadn't had any discussion with a clinician who was caring for them about trials. And remarkably, but also not remarkably, another 50% said participation is disruptive to their daily routine. So it isn't easy to get participants. And then there are the regulators at every level. This is changes in the design and execution of trials with increased number of tasks, frequency, work effort, and execution burden, while investigator payment has not been increasing. Again, this is Ken Caton, who's the director of the drug discovery group at Tufts. So it's because of these things and the large unexpended balance that the GAO found in the NIH trials in 2010, that NIH decided that they needed to operationalize the execution of trials. And that's what the Trial Innovation Network, of which we are one of four partners, Utah, Duke, and Vanderbilt are the other main partners, with Vanderbilt and Tufts taking shares of some of the activities that we've been working on for the last 6 years. And that's the story that Doug alluded to, that I'll tell you a bit about. These next slides are NCATS slides, and it's what NCATS, who was the funder of our ICTR or CTSA, this is their story. Clinical trials are only 10% of NIH budget, which is an interesting number. I thought it was bigger, but it turns out not. And it's in the unexpended budget here of slowly performing trials, poorly executing trials, that NIH ran into trouble with the GAO. In order to speak to that trouble, Francis Collins, as part of his stewardship with us, the American taxpayer, and Congress, launched a new era in clinical trials trust. This is a one-page JAMA article, which is worth reading if you haven't read. This is a diagrammatic picture of what they say. They want better, good clinical practice training, better review of applications for clinical trials within NIH. They promoted the single IRB, which, although its attempt was to speed up, has to date slowed down the starting of trials, although our award has allowed us to define the mechanisms to speed that up, and I'll show you a bit about that in a second. They've required a standard protocol template, an FDA review where needed, and people are becoming enforcing the clinicaltrials.gov registration rules. Those were the main things that they decided to do. And just like pharma, here's NIH funding. They want us to do all those things with no real change in funding. And remember, the clinical trials aren't 40 billion a year. They're down here at 3 to 4 billion. So these are directly from our first year in the Trial Innovation Network. At the time, our program officer was Monica Shaw, who had come from DCRI, been at NHLBI for a decade, and had run the heart failure studies for NHLBI. She felt we were going to build a trial superhighway. I think we're still on a paved road, but here are some things that we focused on operationally. A single IRB reliance model, some standardized contracting, harmonized data collection, streamlining protocols, and taking this organization to research teams and to patients who need more help in engaging in trials, as that survey I showed you suggested. Monica liked the MBA business world, and she said we should be disruptive. I don't disagree, and I showed you an example of how the internet, the cloud, and media can interact to build a core lab for trial that's much more efficient. We focused on operational innovation, not design innovation, and speaking to this problem of not getting trials up and running and not recruiting patients. These are some of the main issues that everybody faces. And some of our solutions that we've put forth in the consults we've done with people from all over the country through their CTSAs have been solutions that we've been pushing. And as you can see, and Doug alluded to it, if you have expertise in CIRB or expertise in contracting or protocol development expertise or recruitment expertise, you can project that expertise into helping others design those particular elements of the trial. And then when you put them together into a sequence, you have a trial. Karen Lane has worked directly on the startup problem. And of course, you have to have a plan before you start. But if you look here at these different colored boxes, she's developed a curriculum for yellow, regulatory training, blue, SIRB, and contracting, which are the three things you need before you can do site activation. That curriculum alone doesn't work unless you have a teacher who helps you execute that curriculum. The teacher is the site navigator at the DCC or CCC, usually the CCC, that helps the coordinators and the study team do the things that are necessary to get the site up and activated. There's a lot of detail here. I'm going to show you a slide you can't read. These are the sequential activities in regulatory yellow, IRB blue, and contracting that need to go on. And the little numbers are the days that it often takes to do this. And a weekly call from the site manager to help the individual who has to execute all of these tasks, identify and overcome whatever the hurdles are, is what we've worked on in order to start the trials on time. And here's a real data about how that works using Karen's curriculum. This is my own trial, Misty 3, Karen's own trial, Nicole's own trial that we thought we were doing a pretty good job on, with the average startup time being 240 days, and there being lots of laggards. Some of this is the University of California Regents, who took 3 years to approve our contract. Our first project in the Trial Innovation Award was to help a neurology investigator with a 60-site PCORI trial. And we helped her get her sites up in about 170 days as a mean. And our second was a heel opiate trial that we got up and running in. The dotted line is our 3-month, 90-day startup goal in under 90 days. 2 pandemic altered trials, one on knee pain and another one in Alzheimer's, were slowed in their actual performance. And this is actual performance, the times the sites started up, because the drug wasn't manufactured and because the pandemic shut down all patient recruitment. In the per protocol version of their performance, when you take that data out, they come in under 90 days as well. This is a pediatric trial, and these are the 2 rounds of recruitment in our convalescent plasma trial, all now in under 90 days when you focus on operational efficiency. In the last 5 years, the Trial Innovation Network has reached out and done 305 trial consultations to the 65 faculty on the 65 CTSAs across the country. We've done 40 comprehensive consultations, which means we've submitted grants to NIH at the end of those comprehensive consults. We've supported the SIRB needs of trials 130 times, a lot of standard agreement support. And prior to all that, in our first year, we did demonstration projects like the one I showed you in the previous slide. This one here, Treat MS, where we help them get started up quickly. We've done during the pandemic, both the opiate and the COVID programs. And this is just Johns Hopkins alone. We've supported 7 distinct trials, and our set of comprehensive consults, which is about 10, 50% of our trials are now up and running, approved through peer review at NIH. So lessons learned from that. Coordination is like research. It takes time and re-communication. Trials require 3 things, and it's not all about the leader. A lot of it is about the consortium, and a lot of it is about the organization. And then finally, I've had this saying over my desk for 35 years: you can't implement without the 7 Ps. Prior planning prevents piss poor performance. And you need to understand the importance of planning and replanning. All trials have 5 hurdles that are the same: plan and start, screen and enroll, make the intervention and share how you're making the intervention, and make sure you're making it the same way. I showed you examples in Clear and Misty where people weren't making the intervention the same way. Collect data, and particularly collect the endpoint, and then analyze and write. The Trial Innovation Network has worked on the first 3 hurdles, and I showed you some data on the plan and start area. We also work on the screen and enroll and the intervention and data sharing. The last 2 are yet to come, and they're important to trial quality as well. A little bit about our group in a bit more detail. These are some of the silos of activities that BIOS has, and developing expertise that's cross-applicable to between trials and to other types of trials has been relatively easy for us to do once we move to this area of expertise organization. This is a partial overview of our portfolio of trials, and you can see that the majority are since 2015, and many with INDs and many that are IND exempt. This is the kind of sequence of activities that are part of a consult that we do for evaluation, planning, and then support. And we bring the patients in at every stage of that design process. This is our current portfolio of trials that we are, these 2 have been started. I showed you about those. And the plasma trials from the pandemic, of course. And these 4 are starting right now, and will go on for the next 5 years. In the plasma trials, we had very generous DoD support, somewhere above $60 million. And we ran 2 trials. This one is still running. This one stopped because the vaccine availability stopped participation. The results are pending. I can tell you the results are null hypothesis results with a point estimate favoring the intervention. And we don't know the results here. We had substantial delays in recruitment as the pandemic waned. Monoclonals interfered with the trial by altering the percent of high-risk participants. The event rates that we predicated the trial on were much lower than the Wuhan data. which is something that happens in a pandemic. The FDA package development, which we're doing for the Department of Defense, is a major undertaking to do that while you're doing the trial. But we've accomplished that. And the results are pending. We need another 100 to 200 patients. I thought the one hard thing would be doing the trial in a year or a year and a half. And it looks like we're going to have this done at the 18-month mark. But what I didn't think about as we went forward, we applied to BARDA and they turned us down. But pharma had a $20 billion subsidy from the federal government. And these are the actual pharmaceutical companies. And the amount of money that BARDA put into vaccine development at each of these. It's hard to compete. It's particularly hard to compete when you can't get your message out publicly. One of the things we've learned is you have to go direct to patients. And one of the hurdles we had was, despite the generosity of the DoD, they cut out our budget for going directly to patients at the critical peak time in the pandemic. We'd be finished if they hadn't done that. And competing trials, which you see here, is really a major issue. And it becomes even worse in a pandemic where communication to patients has mixed messages and there's over communication with mixed messages. So things we learned during the COVID year: hard to do a trial in a year. Pandemic changes create unanticipated changes in trial execution. Direct to participant is critical in a pandemic. And when we have at the very beginning and now at the end, a full public relations support with marketing and a media program, you can go directly to patients. And we're recruiting well now that we have that. Many people are flexible about the trial, but institutions became increasingly less flexible as the pandemic went on. And although it sounds like a lot of money, 70 million is not enough to support when your competitors are executing billion-dollar programs. That's the story of BIOS, and I'm happy to take any questions. I'll leave this thought up here. Some of this guides how we attempt to behave. We don't always, our actions don't always make it as well as this guide suggests. But when you have 60 to 100 people working on multiple trials, there needs to be some group integrity. And these are thoughts that define how we behave. Thank you very much.
Dan, it's great talk. I have a couple of questions to at least start the questions. The first is, I was impressed with the fact that the COVID trials got up and running and patients enrolling within 3 months. And you might just talk about sort of how you took the modules that you have modules for each part of this that you just suddenly plugged in to get it going. And the second is you talked about direct to patient marketing. You might talk about what you did in terms of direct to patient marketing, what platforms you used, what media you used, how you did that in order to improve recruitment, and sort of your estimate of how much faster it moved things along.
Yeah. The first one's pretty easy, the details of which I think are complex and probably should go offline. We use Karen's curriculum. It works. You tell people, you don't give people a list of 100 things to do. You give them 2 things to do each week. And over the 9-week period, they can get up IRB, contracts, and training. The second, how to go to direct to patients. I was pretty stupid about this, just to be clear. My background has been in the first trials, 10 ICH trials, the 5 subarachnoid hemorrhage pharma trials, my early virology trials. I just had to walk into the ICU to find the patients. It doesn't, I didn't have to think much about it. So the first thing we did was we went to a company called AutoCrute that puts your all your patient materials up on the internet, and people can search for your trial. And AutoCrute is motivated, and they get a bounty for each patient you find. The problem was, as good as our materials were, and they were good materials done by professional trial people, both inside BIOS and outside, and with the full help of the IRB and some ICTR help. They weren't compelling. We didn't get many patients. Francis Collins called us on the carpet in August and said, you know, why do you only have 100? Well, actually, we had 200 patients then. And we were the only performing trial because we were up and running and nobody else was up and running. Why haven't you finished your trial? And the people who had NHLBI money said, you know, we're just starting, and that's why we haven't finished our trial. And they took a long time to start. And I said, we're not doing our trial because we're competing with other people and we don't have the patient's attention and we need a national program. And he said, what would it cost? I said, $50 million. And they said, no, thank you. We eventually got most of that out of DOD. And we interviewed 2 public relations companies that do trials for pharma. And we hired one of them, a New York one. They scan the news every day. They find out what people are hearing. And then they go to the venues that individual people likely to be in the trial follow. And they get our story and our materials out there. And they built a much better website that fell down into a call center where people who saw our stuff on the internet could call after going through a screening instrument to see if they were eligible. And the yield on that now is 25% of the people who come to the call center enroll in the trial at Hopkins. It's a pretty nice system. Costs a pant load of money. That's, you know, when we did the Misty and Clear studies, when we got to a phase 3 study, we had to have a plan to spend $25 or $30 million over 5 years. A plan to spend $70 million over a year is a much bigger task to execute. And you need help. And quite honestly, we all know the mixed message story. And that was our solution. Unfortunately, DOD stopped our use of that capability from December until May. And we missed out on the peak part of the performance in terms of the pandemic, the peak incidence of the pandemic.
Okay. Other questions from people?
Hello, good morning. Thanks a lot for the nice talk. And I have very general question. How difficult would you say it is to obtain funding for conducting a clinical trial, as the success cannot be predetermined? So
Yeah. So I think I hear, thank you for the question. Is it Ishta?
yes.
It's a good question. I hear 2 questions. One is how difficult. It's obviously difficult. 10% of all grants get funded at the NIH. So that's 1 in 10. And it's probably a little worse for clinical trials. The second question I hear is, how do you actually get something funded when you don't have clinical trial data? And in that question, I think that's where you use open label data collected around a hypothesis that more likely than not is a hypothesis in a causal pathway of the disease with a biomarker. And we started these ICH trials with essentially free data collected about a simple endpoint, mortality, in sets of 30 patients. And we used as a biomarker the volume of the clot and the location of the clot, which we had a robust algorithm that predicted mortality, and we showed that we altered that prediction of mortality. That was the preliminary trial data that we used. And I think it points out something that your question was addressing. There's some circular logic here. You can't do a trial without having done a successful trial. That's been our solution to that circular problem.
Dan,
So
hey,
you-
so Neil Goldenberg, that was a wonderful overview, and the philosophy was very much appreciated along with it. I wanted to ask you your impressions about the contracting side of things in site startup efforts. I think, of course, with SIRB, you can argue that maybe there's some more complexity, but I think in general, a lot of the times the rate limiting step can be the contracting piece.
Yep.
And are universities in general well equipped to handle multi-center trial contracting if they have multiple PIs doing it at the same time? Or does one, whether they use a BIOS or a similar org at a university, do they need to have internal staff who are liaisons with grants administration to help dog the process and understand the process and work with grants and contracts?
The second, okay. Grants and contract offices are overwhelmed at every institution. And, we have great faculty who put in lots of grants, but 90% of what they put in doesn't get funded on the first time. It can be as high as 30% if you persist, which is another answer to the question that was posed previously. In terms of getting the contract through at your consortia, obviously a 70-site consortia is a big problem. And we've worked backwards from performing on that problem to how we do things. And there has to be both an internal finance officer and a lead program officer for that particular study that understands the site issues and contracting. And you have to listen to your site, which I mentioned in the talk. Now, how to listen to the site and how to frame what you're doing is what's important. And Karen Lane and I, in dealing with the disruptive issue of doing surgery on patients with ICH, came up with a formula that's worked pretty well. And that is use the Hopkins PRA system, which evaluates what is clinical care and what is research, but then take it to the Medicare reviewers. There are 5 national Medicare reviewers for the, this doesn't work in pediatrics, obviously. 5 national reviewers for payment determination. So we took our PRA, which we had carefully developed here at Hopkins, and we submitted it to the 5 regional Medicare reviewers and asked them, did they approve it as well? And they did. So then when we went to our 70 sites, only one of the 70 happened to be the Cleveland Clinic, said this was not correct, and they couldn't do the trial because they thought that we were going to be asking them to pay for trial activities. They had never performed in any trial I'd done, so it wasn't a big loss. But that had convinced all the other sites to go forward. When I had the individual site-by-site discussion with the Cleveland Clinic, I said, "You're welcome to your opinion, but the protocol has been approved by your reviewer, and there are 2 other sites in Ohio that are doing the trial and getting paid for the routine clinical care part of the trial." That kind of strategy, going to a third party, getting a third party approval, gives your people who are dealing with folks like us at all the other sites some legitimate third-party review to push forward the trial, which is going to disrupt care, and it suggests that there's not going to be a problem with payment.
Thank you. That's really helpful.
So, Dan, my last question to you is just to talk a little bit about your work with Strides, and because it seems to me that that's a huge improvement in sort of from a manual reading center to a very quick reading center solution for interpretation of data and for estimating the outcome, which was the amount of hemorrhage present.
Right.
You said it was expensive. I don't even know what you mean by expensive, so maybe you can give us a ballpark figure of what it costs to do that, but just a little bit about that interaction, because I think that's a real opportunity to make outcome determination much more efficient in trials.
I guess I'm not sure where I said it was expensive. It's expensive to do with human beings and through the routine clinical mechanisms. Most sites would charge $15 to $30 to make a CD of a DICOM file, which a coordinator would have to go and bring to her computer, and then either mail to us or send over the internet. And prior to that, most sites made a silver-based copy of a radiologic DICOM CAT scan film, which was very expensive, and mailed it to us. And then we would originally we had a radiology technician do the segmenting, a little bit the way the Retina Reading Center worked at Wilmer 30 years ago. Things have gotten cheaper. We now have, over a series of 10 trials, we have every brain coordinator in the country pretty much trained on going to their EMR, grabbing the DICOM file, and dragging it onto their desktop, and then going to our website and dragging the file onto our ECRF. And then the next step was the Strides program, in which we took probably 10 to 15 publications that we'd done on how to measure that hematoma. We had automated much of the process already, and others were doing it. We took that process and defined some steps. The two major ones, or the three major ones, being identify all the HIPAA non-compliant elements in the file and strip those out, and strip them out with much more stringency than coordinators were doing. And the second was identify the structure of the individual brain as a template, and then find the lesion, which has a density contrast, So you can write formulas to do that. And with the rapid computing power on the cloud, we took probably the 5 programs that did those tasks and combined them into a single program. So now the coordinator drags the DICOM file to a cloud website where those 3 main activities, strip, identify the individual's brain and some landmarks in it, and then circle the lesion. The circumference times the width of the slice equals a volume, and then you add up the volumes from all the slices. And just like you can order something on Amazon, you get the core lab back in 100 seconds. The cost of that is 1/10,000th of the cost of the original system. And so the real question is, and this is why we're framing it as a core lab, how do you push that forward? Do you take probably $1 million of the $25 million on the MISTI grant was this core lab. We can now do it for 100,000. And the question is, do we make the grant $1 million cheaper, or do we take that $900,000 savings and put it into screening many more patients at the sites to make the trial go faster? And I'm going to favor the second. I want to take the cost advantage and putting it into doing our job of finding patients faster. Does that answer your question?
Yeah, I think so. So we are over by a few minutes. I want to thank everybody for attending. I want to thank our speaker, who did a great job. And we're done. If anybody wants to stay on for a minute or two, Dan, I don't know if you have a couple minutes to answer any sort of smaller questions, but if not, I'm
happy to answer more questions if people have them.
Yep. We are finished the seminar per se, and looks like people are starting to stream out the doors.
Do you have any other questions, or does anybody else?
Looks like you are incredibly clear, so
good.
Great.
Good. Have a good day, everybody.
Take care.
Bye-bye. Thank you, Taylor.